Welcome, AP Biology students. This is our chapter on respiration, both anaerobic and aerobic respiration. So, what you need to know from the chapter, these are the basics of what you should understand for the AP test. You should know the summary equation of cellular respiration, differences between fermentation and cellular respiration, the role of glycolysis in oxidizing glucose to two molecules of pyruvate, the process that brings pyruvate from the cytosol into the mitochondria and introduces it into the citric acid cycle. And then finally, how the process of chemiosmosis utilizes the electrons from NADH and FADH2 to produce ATP. In open systems, cells require energy to perform work, chemical, transport, and mechanical work. So in order to do that, we need to have energy. And we know that energy flows in the ecosystems of sunlight. That solar energy is harnessed by plants in the chloroplast. And ultimately, what will happen is they'll use photosynthesis to transform it into chemical energy, and oxygen is released as a byproduct. Cells use some of the chemical energy in organic molecules to make ATP. And you know ATP is the energy molecule of the cell. AT powers most cellular work. And then ultimately, during every energy conversion, it is not 100% efficient that we see in the laws of thermodynamics. So we do lose some energy in the form of heat. So energy leaves as heat. So ultimately what we're going to see here is we're going to see a catabolic pathway. And in the catabolic pathway, complex organic molecules are going to be broken down into simpler waste products with less energy. And some of that energy is going to be used to work and dissipate as heat. Ultimately, this is the opposite of what we saw in photosynthesis, which was an anabolic pathway. Because energy is being released in respiration, we call that an exergonic reaction. And it's ultimately what you had as a product and reactant for photosynthesis is now reversed and going to be your reaction for cellular respiration, where you have a glucose molecule, C6H12O6, plus oxygen gas, 6 molecules, and then you're going to produce 6 water molecules, 6 carbon dioxide molecules, and energy in the form of ATP. Also, you have heat energy there. Photosynthesis is an endergonic reaction, and therefore it required that energy. So you could see the relationship here between autotrophes and heterotrophs, and how photosynthesis and respiration are, although very different, do have the same molecules, whether it be as a reactant or as a product. Like in photosynthesis, we do see redox reaction here. There's oxidation reduction. Again, oxidation is the loss of an electron from a molecule, and reduction is going to be the gain of an electron to a molecule. And there we see the oil rig, or Leoger, as the analogy. So what happens is we see those molecules being oxidized and those molecules being reduced. And here's an overall summary of what's going on there. So you can see how you have the electron flow in this particular equation here from reactant to product, where you take methane plus oxygen gas and make carbon dioxide, energy, and water. Energy harvest. Energy is released as electrons fall from organic molecules to oxygen. Food from glucose from food is going to form NADH in the electron transport chain, and we're going to have oxygen. And ultimately, the role of oxygen there is going to be very important. And that's why we call it aerobic respiration. We have coenzyme NAD+, and that's going to serve as an electron acceptor. NAD plus picks up 2 electrons and also picks up 2 hydrogens, and you form NADH. NADH is going to carry electrons to the electron transport chain. In the electron transport chain, we're going to see the transfer of electrons from those molecules to oxygen, and ultimately the end product there we get is water. We also have that release of energy. So if you look at the NAD plus as an electron shuttle, here you can see nicotinamide oxidized form plus two hydrogens. And then you also have the enzyme, which is an enzyme called dehydrogenase. That's going to help with that reaction to convert NAD plus to NADH. So you can see the two electrons going in plus the hydrogen. There you form the nicotinamide, which is the reduced form, and then you get another hydrogen there. In the electron transport chain, remember, an electron transport chain is anything where you have a series of membranes, and it's going to transport those high energy electrons and use the energy that comes out of those electrons to move hydrogen ions. You can see in the diagram to your right that the free energy is on the y-axis, and then you have the cellular respiration process down below as what it's illustrating there. So in this case, ETC is not taking place in the chloroplast. It's going to be taking place in the inner membrane of the mitochondria. So the stages of cellular respiration are glycolysis. Glycolysis simply means the splitting of sugar. You have pyruvate oxidation plus the citric acid cycle, which is known collectively as the Krebs cycle. And then you have oxidative phosphorylation. Oxidative phosphorylation is the combination of the electron transport chain and chemiosmosis. If we look at that, you can see that glycolysis is where we have substrate level phosphorylation. In substrate level phosphorylation, we have ATP being made there. We have substrate level phosphorylation occurring in the citric acid cycle. So in both those cases, we see one occurring in the cytoplasm, and then the Krebs cycle and the oxidation of pyruvate and the ETS are all going to take place inside the mitochondria. And then we get oxidative phosphorylation. Oxidative phosphorylation is what we see with ETS and chemiosmosis. So let's look at the first stage of cellular respiration, known as glycolysis, split sugar. It's believed to be an ancient pathway. Early prokaryotes had no oxygen available. So glycolysis takes place in both aerobic and anaerobic conditions. It occurs in the cytosol of a cell, and it's going to partially oxidize glucose to form two pyruvate molecules. So you're going to go from a 6-carbon glucose to 2 3-carbon pyruvate or pyruvic acid. Ultimately, the net gain there is going to be 2 ATP and also get 2 NADHs. And the net gain is not the same as what the output of ATP is going to be. The output of ATP will be 4 because initially in glycolysis you put in 2 ATP. Your net gain is actually what you're going to be yielding. So you get 2 ATP total. We also do generate some water in glycolysis, and it's in no oxygen required process. So in the first step, we have that energy investment stage, which I was just explaining about. The cell is going to use that ATP to phosphorylate compounds of glucose. In the second stage, energy payoff stage, 2 3-carbon compounds are going to be oxidized for each glucose molecule, and you're going to get 2 net ATPs that are going to be produced by substrate level phosphorylation. Ultimately, then the 2 molecules of NAD plus are going to be converted to NADH. What is substrate level phosphorylation? Ultimately, it's a process that is going to generate a small amount of ATP. Phosphorylation is enzymes are going to transfer a phosphate to other compounds. So if you have a phosphate group plus a compound, you could say that's been phosphorylated. In this case, ADP is going to pick up that free phosphate group, and you're going to form ATP. You could see the process there to which it is done. So if we look at glycolysis all together, here's our overall energy reaction figure. And what we see in this part of the metabolic pathway are two steps. In the first step, we call it the energy investment phase. And that's because we need to have an input of energy to kind of get the reaction going. So ultimately, glucose is going to come into the cell. And in order to keep glucose into the cell, we need to invest some energy there. And what happens is you have an enzyme called hexokinase. Hexo meaning it's a 6-carbon sugar, so hexo, hexagon, 6. Hexokinase is going to take that glucose molecule and phosphorylate it to glucose 6-phosphate. Then you have phosphoglucoisomerase. Phosphoglucoisomerase, if you break down those words, it's a glucose molecule with a phosphate group attached to it, and you're going to change it. Isomerase, remember, isomers are molecules of the same molecular formula, but their structures are different. So glucose 6-phosphate is going to be converted to an isomer called fructose 6-phosphate. And when that happens, we have the enzyme phosphofructokinase come in, and it's going to take another ATP molecule, and you're going to get fructose 1,6-bisphosphate. Ultimately, then, that is going to split into two molecules, and you see them different there. They are isomers of each other, but we're going to look at it as glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is the molecule that you know of from photosynthesis. That's G3P that is going to come out there. The other one is dihydroxyacetone phosphate. That's an isomer form. So in this first overall step, the basics of what you need to understand here is that glucose is going to be converted to fructose 6-bisphosphate or fructose diphosphate after two ATP molecules come in. That kind of stabilizes it, and then it is split using the enzyme catalase to form two G3P molecules. So energy is invested. Now we need to take those two G3P molecules and have an energy payoff phase or an energy harnessing phase. And in the second step, each of those G3P phosphate molecules are now going to be converted to pyruvate. So if you track one going through, ultimately what you get is one G3P molecule is going to be converted to pyruvate. You could see all the intermediate molecules that are in there and the enzymes that play a role from converting one reactant to another product or intermediate until you end up with the final result of pyruvate. So if you follow it through, just one molecule, you get out one ATP, one NADH, and you get out 2 ATP for the first part, and then another 2 ATP for the second phase. So glucose, glyceraldehyde 3-phosphate gets converted to the intermediate. You get one NADH out, and you get 2 ATP out to end up with pyruvate. Then the other G3P molecule is going to come through. You get another NADH out, another ATP initially, and then right before you convert to pyruvate, a second ATP. So total getting out of there is a net yield of 2 ATP and 2 NADHs, even though you have 4 ATP coming out. You only have a net yield of 2 because initially in the energy investment phase, you have 2 ATP. And I'll go over this again in class to explain that further. Now, what happens after glycolysis is going to depend on the availability of oxygen gas. So glycolysis, in summary, glucose gets converted to pyruvate, 2 ATP and 2 NADH and 2 water molecules come out. Now we have pyruvate, the oxidation of pyruvate plus the citric acid cycle. So oxygen gas is available. And in order to understand what's going to happen here, we need to take a look at the structure of the mitochondria. So the mitochondria, like the chloroplast, has been once thought to be a organism of its own, having its own DNA double membranes. Those are all evidence that it has evolved um, as the organelle into the eukaryotic cell, but was once a free-living prokaryotic cell. And ultimately what we see here is that outer membrane and the inner membrane, there's a space between there called the intermembrane space. And the inner membrane is folded. And when the inner membrane folds, you form these structures called cristae. Remember that in biology, when we talk about structure meeting function, if you look at the structure of the inner membrane, all those folds are going to increase the surface area for a reaction, all right? In this case, it would be the ETS system, because we're going to see electrons traveling along that inner membrane, because the ETC requires a membrane. In the inside, you have the mitochondrial matrix. So the mitochondrial matrix is going to contain the enzymes and the materials required for the citric acid cycle, or the Krebs cycle, as you may also know it as. So if we look, pyruvate comes in from glycolysis, and ultimately what we see happening is pyruvate gets converted to acetyl CoA. That happens when you have coenzyme A coming in. So acetyl CoA is going to be the end product, and ultimately in that transition step, you get out an NADH molecule, you generate some carbon dioxide gas, and you form this molecule right here. Acetyl CoA is now going to be able to enter into the citric acid cycle. Acetyl CoA will come in, coenzyme A will come off, and ultimately what's going to happen here is in the citric acid cycle, you produce very small amounts of ATP. But what you do see being produced here is a little bit of carbon dioxide and a lot of electron carrier molecules. So we get out 3 NADHs, we get out an FeDH2, we get out an NADH up here. This process up here, the transition step, is separate from what's going on down here. And all of these electron carrier molecules are going to go to the electron transport chain where we're going to produce the bulk of ATP. So let's look at pyruvate oxidation, or that transition step. Pyruvate is going to come in from the cytosol into the mitochondria via transport protein, and ultimately we get out carbon dioxide gas, NADH, coenzyme A comes in, and we form acetyl CoA. This is going to happen for every pyruvate that is formed at the end of glycolysis. And if you recall, that is 2. So each pyruvate will go through this step. So pyruvate to acetyl CoA is going to be used to make citrate. Remember, citrate is the first stable product of the Krebs cycle, and that's why we give it its name, the citric acid cycle. Carbon dioxide gas and NADH are produced. In the citric acid cycle, this occurs in the mitochondrial matrix. The acetyl CoA is going to form citrate, carbon dioxide gas is going to be released. You're going to have a net gain of 2 ATP, 6 NADH molecules, and 2 FADH2 electron carrier molecules. ATP is going to be produced by substrate level phosphorylation. So here are, after the transition step, here is the acetyl CoA coming in. So acetyl CoA comes in, it binds to oxaloacetate to form citrate, which is the first stable product of the Krebs cycle, which is also why we call it the citric acid cycle. The next step, we form some intermediate molecules. So we form isocitrate, ketoglutarate, all the way down to succinyl CoA. Now, if we look at this, the conversion to citrate down to here is where we generate some CO2. So we have some loss of carbon there. We have some loss of carbon here. We form some NADH molecules here and here. And then when we convert succinyl CoA to succinate, what happens is we get substrate level phosphorylation. So ATP picks up a phosphate group to form ATP. And you can see here where that phosphate group is coming in. All right. Now, when you go from here to here, you're going to form electron carrier molecule FADH2. So from succinate to fumarate, you get FADH2, and then fumarate gets converted to malate, and malate is going to be converted back into oxaloacetate. And when you do that conversion, the eighth step, you get another NADH out. So that is one turn of the cycle. You do have a second turn because you have the second acetyl CoA from the second pyruvate glycolysis coming through. And the same thing's going to happen. So acetyl CoA is going to form oxaloacetate, bind with oxaloacetate to form citrate. And then the conversion all the way down through, you get out another NADH here, another NADH here, lose some carbon to CO2, you get FADH2 here, you get substrate level phosphorylation occurring here to form ATP, and you get another NADH out here. So in total, per one turn of the cycle, we get 3 NADH molecules, 1 FADH2, and 1 ATP. Total, we get 6 NADH molecules, 2 FADH2s, and 2 ATP because you have 2 pyruvate at the end of glycolysis. So the summary of the citric acid cycle is 2 pyruvate being converted to 2 acetyl CoA. And ultimately, the outputs are 2 ATP and 8 NADH, 6 CO2, 2 FADH2. Now, I don't know if this will work. Let's try it out here. We'll have to come back to that. The last step, oxidative phosphorylation, is a combination of the electron transport chain and chemiosmosis. The electron transport chain is going to occur in the inner membrane of the mitochondria, and it's going to produce 26 to 20 ATP by oxidative phosphorylation via chemiosmosis. Chemiosmosis is going to utilize the hydrogen ions pumped across the inner mitochondrial membrane. As hydrogen ions diffuse through ATP synthase, you're going to get some energy. So if we look at the electron transport chain is a collection of molecules, much like we see in the chloroplast with photosynthesis, that are going to be embedded in the inner membrane of the mitochondria. Here we see them. They are the multi-protein complexes. So those tightly bound protein and non-protein components are ultimately going to allow for the alternation between reduced and oxidized states as electrons are going to be accepted and donated. This does not make ATP directly. What happens is energy is going to fall from the electrons from food to oxygen. So why is oxygen required in this process? It is because oxygen is going to be the final electron acceptor. So in aerobic respiration, oxygen gas is the final electron acceptor. And when oxygen picks up those electrons, it also picks up hydrogens, and there we form water. And we know that we have water as a byproduct of cellular respiration. As electrons move through the ETS, proton pumps move hydrogen ions across the inner mitochondrial membrane. So here you can see the electron carrier molecules, NADH and FADH2, that were all made during the process, coming in the processes of glycolysis, the transition step, the Krebs cycle, all coming in and dropping off their electrons. And they're passing through these protein complexes and using these protein complex electron carrier molecules to move those hydrogen ions from the mitochondrial matrix into the intermembrane space. Remember, the intermembrane space is the area that is between the outer and inner membrane of the mitochondria. Chemiosmosis allows for energy coupling mechanism. So in chemiosmosis, you now have that electrochemical gradient or that proton gradient across the membrane, and that's going to drive cellular work. So the proton motive force is going to use protons, the H plus gradient, to perform work. And that work is going to be aided by ATP synthase, the enzyme that makes ATP. So here you can see as the hydrogen ions come in, here you have that rotor, the hydrogen ions come in and it's turning this catalytic knob to help with the production of ADP plus a phosphate to form ATP. All right. So the use of energy from the proton gradient and the flow of hydrogen ions back across the membrane is what helps drive this process. So here you can see the electron transport chain. Here you see chemiosmosis. Collectively is why we call that oxidative phosphorylation. So our end product is water and ATP. Coming into this process, we have NADH and FADH2s. So oxidative phosphorylation uses chemiosmosis to generate ATP. It uses energy in redox reactions. Redox reactions of the ETC system, in which electrons are going to be passed down energy levels to the final electron acceptor, which is oxygen, and oxygen will ultimately accept those electrons and hydrogen ions to make water. Oxidative phosphorylation also uses chemiosmosis, which couples the protein gradient called the proton motive force to drive hydrogen ions through ATP synthase to produce ATP. Ultimately, electrons pass down energy levels as the hydrogen ions pumped from the matrix to the intermembrane space. They are going to help with that proton gradient. So overall, as we see, ATP yield per molecule of glucose at each stage of cellular respiration, we could see that you get about 30 to 32 ATP. Now, I would caution you that it does vary. Some authors do say 34 to 36, 30 to 32. ATP variations do occur. But ultimately, where are we getting the numbers for here? We get 2 ATP out of glycolysis, we get 2 ATP out of the citric acid cycle, and we get about 26 to 28 ATP out of the electron transport chain, which yields about 30 or 32 ATP maximum per glucose molecule going into aerobic respiration. So cellular respiration also includes anaerobic respiration. Anaerobic respiration is going to generate ATP using other electron acceptors besides oxygen gas. The final electron acceptors include sulfate, nitrate, and sulfur, and it's going to produce H2S, hydrogen sulfide. We have two different organisms that could carry out these processes. You have obligate anaerobes and facultative anaerobes. Obligate anaerobes can't survive in oxygen. Facultative anaerobes are going to make ATP by aerobic respiration when oxygen is present, or switch to fermentation when no oxygen is present. So obligative anaerobes can't survive in oxygen and ultimately only function in anaerobic processes. They're obligated to an anaerobic condition. In facultative anaerobic respiration, those organisms could do both. They could switch between aerobic and anaerobic respiration. An example of facultative anaerobes would be human muscle cells. Fermentation utilizes glycolysis, and the importance of fermentation is that regeneration of NAD+. Glycolysis with oxygen and glycolysis without oxygen. So ultimately, you know already that after glycolysis takes place, the availability of oxygen gas is going to determine whether it's going to be fermentation or respiration. In fermentation, fermentation allows glycolysis to continue by regenerating NAD+. It occurs in the cytosol, no oxygen is needed, and you could create ethanol and carbon dioxide gas or lactate as a byproduct, depending on the cell or the organism that is carrying out the process of fermentation. Ultimately, the only amount of energy you are making is the 2 ATP from glycolysis. The reverse of that would be respiration. You get a release of energy from the breakdown of food with oxygen. It occurs in the mitochondria. Oxygen is required because it is that final electron acceptor. And in doing so, you're going to produce carbon dioxide gas, water, and up to about 32 ATP molecules. So you could see a larger organism such as ourselves needs respiration in order to produce that amount of energy for the functioning of our cells. But something small like a yeast or a bacterium or another type of small anaerobe could use the process of fermentation. And those 2 ATP molecules generated from fermentation will allow that organism to function cellularly because there isn't a demand for a large amount of energy as you would see in the human body. So the two basic types of fermentation are alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation, pyruvate, is converted to ethanol and oxygen gas. Example organisms are bacteria and yeast. In lactic acid fermentation, pyruvate is going to get converted to lactate. An example where lactic acid fermentation takes place: fungi, bacteria, human muscle cells. If you think of baking bread or making bread dough, you allow the dough to rise. It's an alcoholic fermentation process, and what causes the bread dough to rise is the product of alcoholic fermentation, which would be carbon dioxide gas. And ultimately, why we don't get drunk when we eat a piece of bread is because during the baking process, then we burn off that alcohol or that ethanol. So alcohol fermentation is used in baking, brewing, and winemaking. Lactic acid fermentation is used to make cheese, yogurt, acetone, methanol. A note that lactate can build up. It does not cause muscle fatigue or pain, but it does go back to that liver to regenerate back to pyruvate. So various sources of fuel. Carbohydrates, fats, and proteins can all be used as a fuel for cellular respiration. And overall in the diagram, you could see how those particular biomolecules would come into play throughout the steps of that process. So monomers enter glycolysis or the citric acid cycle at different points. A key thing there is an enzyme called phosphofructokinase. Phosphofructokinase, allosteric enzyme that controls the rate of glycolysis and citric acid. So remember, as an allosteric enzyme, ultimately what we see there is if phosphofructokinase is going to convert that fructose 6-phosphate into fructose 1,6-bisphosphate. So if we start to generate a lot of ATP molecules, ATP can come in and bind to a point on that enzyme other than the active site and halt that process, and it will stop glycolysis from continuing. Or what we see is if we start to produce a lot of citrate, citrate could come in and bind to phosphofructokinase at the allosteric site and inhibit the process from taking place. So there's that negative feedback loop mechanism to kind of maintain the production of ATP in the process. So it's inhibited by ATP and citrate, and it's stimulated by a molecule known as AMP. AMP plus a phosphate plus another phosphate is ATP. So respiration in the big picture, energy comes in, glycolysis in the cytosol, substrate level phosphorylation. Glycolysis, aerobic respiration or cellular respiration with O2 takes place in the mitochondria. You have pyruvate oxidation, the citric acid cycle, and substrate level phosphorylation. ETC, chemiosmosis, oxidative phosphorylation. In fermentation, you have glycolysis, oxygen is not available, so it's going to be an anaerobic process. And that will be fermentation, which takes place in the cytosol. One type of fermentation, you can produce ethanol and carbon dioxide. In another type of fermentation known as lactic acid fermentation, you could have animals. It takes place in animal muscle cells. So if we look at the glycolysis and citric acid cycle, here you can see the inputs and outputs in summary. And ultimately here you can see oxidative phosphorylation as a summarization as well. So that's it for our chapter on cellular respiration. I know it was a long lecture, but I want to get things moving here so we can move right along in AP biology. We will continue with the labs for both photosynthesis and cellular respiration and do some discussion in class regarding this process. Have a great day and thank you for listening and taking notes.